What I'm hoping to do here is walk you a little bit through the process that we went through as we started partnering with Element Bioscience, and then hopefully demonstrate how the AVITI system seamlessly fits into the 10x workflows that we have been establishing over the last few years. Single cell has truly transformed our understanding of human biology. When you move from bulk sequencing to single cell sequencing, you can truly uncover the complexities and the heterogeneities that are hidden behind complex tissues such as cancer or brain. And so what you see on the right-hand side is a typical representation of a single cell or of a sample that's been sequenced in the single cell space, where each dot represents a single cell and each cell is clustering within a specific region based on its transcriptional profile. What has also become very clear is that single cell sequencing and single cell assays have become more cost effective as well as more robust and really kind of accelerated beyond just the one modality readout gene expression. So really this multi-omic space. And as a result has opened up to many, many more labs across the globe. At 10x, there's a number of assays that we have pushed out over the years, both in the single cell space as well as the spatial space. And what all of these assays have in common is that they produce an NGS sequencing ready library that is sequenced before the data analysis and the data visualization. And this is where Element Bioscience comes in. As a researcher, when you start thinking about setting up your single cell experiment and sequencing it, there's a number of elements, or I guess no pun intended, a number of factors that you should really consider as you start designing your experiment and then also sequencing it. The first one is flexibility, and I kind of think about this in many different flavors. You may be able to just get started with single cell sequencing, and the very first time you put your sample on a sequencer, and you may have to actually QC it first before you go full in and sequence it to application depth. You may also want to actually sequence as much as you can from a specific sample and really go deep, as in pooling multiple samples together. The other way I think about flexibility is having multiple applications, right, that may require you to set up the experiment slightly differently. One library may require a different setup than the other, but you want to sequence them all in one go. Throughput is the second factor here, and that's kind of a combination of both cell throughput and sample throughput. In single cell, I like to think about the number of cells that go in per sample, but then also the number of samples that go in per experiment. And both of these combined kind of yield into the number of sequencing reads that you require per experiment. The AVITI system is designed exactly for that type of application, because you can now think about sequencing a single sample that has thousands of cells. Or you can think of having multiple samples pooled together where each sample only represents about 2,000 to 3,000 cells. Each flow cell, depending on sequencing depth, really accommodates well over 20,000 cells per run. And if you can really maximize the throughput by combining both of these flow cells in one sample. High quality is the third piece. And that's kind of a function of the way our library constructs are built. So all of our assays include a 10x barcode, and that 10x barcode in the single cell space is required for you to assign a single cell. In the spatial side, it's required for you to assign a specific spot on a slide. We have a UMI sequence, that's a unique molecular identifier, and that's used for us to count individual transcripts. And then we have the third sequencing species, which is the transcript itself, which we use to obviously map back to our reference. And so you need high quality reads from all three of these species, not only to guarantee a successful experiment, but also to actually maximize the biological insights that you get from your sample. And then the last one is economy of scale, and that's kind of going into the application space. So as you think about sequencing within CRISPR screening or immunology screens or any screens where you need a lot of samples or not of cells, you can only do this if the sequencing portion of this experiment is kind of an economy of scale where it becomes more routine. And so the AVITI system provides such a solution. It opens up the NGS space to really a lot of people beyond just a very small core, and it has a unique chemistry that enables the combination of accuracy, low cost, as well as operational efficiency. And so I got a demo yesterday actually of this instrument, and I was pretty impressed by the by the idea to have these two independent flow cells. So, the idea of thinking of this as almost two sequencers in one unit makes it very obvious why this is so flexible, right? So you can think about setting up the first batch of libraries that come in, set them up. You need data right away, and then setting up the next set of libraries that come in maybe a day or two later. You can also think about having one sample that you just want to split across both flow cells because you have 60,000 cells that you want to sequence down to the maximum sequencing depth. Each flow cell gives you 800 million reads, which for an average sequencing run can accommodate up to 20 to 30,000 cells on a flow cell. You have best-in-class Q30 scores north of 90%, and you really have the ability to set up your experiment such that the libraries or the sequencing kits that the AVITI system provides basically accommodate 3' our 5' solution and our visium solution. Now, for those of you, most of you I see here are probably have seen the insights of this instrument, but if you haven't, I would definitely encourage you to take a look at these demo units. The setup of these two independent flow cells really allows you to think about plexing multiple applications together. You can think about setting up experiments at individual days or time points, and you can really think about the actual reach and maximizing the input or the output of your single sample. And so the sequencing kits really provide a solution where it's low run cost, it's low cost per sample, and it's across a range of applications, as you can see here. Just yesterday, I believe, Element Bioscience released their a new sequencing kit configuration, which is really tuned for applications such as single cell sequencing that really require fewer reads overall per run, but give you the maximum number of reads to basically accommodate the entire range of our portfolio. And so that 1 out of 550 cycle kit has the ability to read through all the sequencing species that I mentioned earlier, gives you the full insight into the transcript, and really gives you that the flexibility, as you can see on those flow cells, to run anywhere between as little as 2,000 cells at maximum sequencing depth to north of 60,000 cells at a lower sequencing depth. The workflow on the 10x side is pretty straightforward. So you have your sample on the single cell side that you dissociate. You load that into our microfluidics. You run it on the instrument. You then generate a library outside of the instrument, and then you use that library that you then fit into the AVITI system. The AVITI system produces FASTQ files, which are then used as input for our Cell Ranger, our software pipeline. And that's basically a plug and play fashion. For anyone that's been using our assays in the past, likely wouldn't even feel a difference, especially if they are submitting their libraries to a core. There's no special requirement for our customers to change the workflows or the user guides. It's really what they have in hand is plug and play with the AVITI system. And so the way we are sort of thinking about this Compatible Partner Program is to not only establish compatibility, but also to really validate it in such a fashion where both the partner that we work with as well as 10x can support our customers. And so the intent holistically is for us to really open up the field to not have our customers and your customers can be constrained in the way they can think about setting up their experiments. And so what we typically do is we have a benchmark sample, in this case human PBMCs, that we had sequenced at different throughputs, so 1,000 cells and 10,000 cells. We ran these in 2 replicates each, and then we generated a library with our 3' kit to then sequence on the AVITI system. The raw data then gets sent back to 10x. Our R&D team looks at that, validates this, and benchmarks this against a control that we have a pretty good understanding on how it behaves. And so as part of this program, we also documented this and produced an application note, which highlights some of the data outputs as well as the actual performance. And so for those of you who haven't seen the application note, also encourage you, since I'm only flashing a few highlights here. The first one is throughput. So you see this here, those two different samples. On the left-hand side, that's the 10x, it's the 10,000 cell sample. And on the right-hand side, it's also human PBMCs, but only 1,000 cells. So you can already appreciate the type of complexity that you can achieve as you sequence more and more cells. And that kind of goes hand in hand with the table below, where as you sequence more cells, you don't require as much sequencing depth. And as you sequence fewer cells, you may want to sequence a little bit deeper to really understand those rare cell populations. So that AVITI system, again, gives you that you can think about the design of this particular mode on one flow cell, and then you can even expand that to 2 flow cells. This one kind of demonstrates the accuracy, obviously. So we ran each of these assays, each of the samples in replicates. And so what you see here is the scatter plot on the left-hand side with an RQ score of 0.999. So obviously virtually identical. The plots on the right-hand side are examples of how much each of those replicates overlap within the cluster space. And then also below, you see in panel D, some of the key markers for some of these human PBMCs are lining up between both replicates, much like we expected. And so, the result was less surprising to us, as we have seen in initial results before we even engaged into the full validation, that all the key metrics that we looked at have passed. Obviously, we have very tight specs internally that we really set high, so that there's a high success rate for anyone that wants to run our libraries on the various sequencers. And all the performance metrics that we typically look at is gene sensitivity, the ability to capture individual cells, the ability to map the transcript back to the reference, and the ability to form clusters such that we actually identify our cell populations. All these key metrics have passed. The AVITI system is compatible with our 3', our 5' solution, which is our immune profiling solution, as well as our visium spatial gene expression solution. And so we're super excited. We here on the 10x side have heard a lot already from our own customers on how the system works, and whether or not we can help them get started on sequencing their libraries on there. And I'm expecting that we're going to have more and more of these conversations moving forward.